Hello everybody! Today we're going to be talking about the basics of cell communication. So there are three types of basic cell communication: long distance, short distance, and direct cell-to-cell communication. Long distance involves plant hormones and animal hormones. Short distance involves neurotransmitters, yeast mating, and quorum sensing. Direct cell-to-cell communication involves plasmodesmata in plants, gap junctions in animals, and contact inhibition, as well as immune cells in animals. The first type of cell communication I'm going to be discussing is long-distance cell communication. This involves plant hormones and animal hormones. In plant hormones, hormones may travel in vessels, but often travel from cell to cell or by diffusion in the air. An example of this would be ethylene, which ripens plants. And this is considered a positive feedback mechanism because in the presence of more ethylene, ethylene is produced. So it's that cycle of continuously producing ethylene. In animal hormones, endocrine cells release hormones into the circulatory system where they travel to target cells. An example of this would be insulin. This hormone is released by specialized endocrine cells and travels to all cells that are in need of glucose. And hormones like this are able to regulate positive and negative feedback mechanisms. These two examples are forms of long-distance cell communication because the signaling molecules, being the hormones, travel a long distance to communicate from cell to cell. The next form of cell communication is short-distance cell communication. This can be shown in neurotransmitters, yeast mating, and quorum sensing. In neurotransmitters, the presynaptic membrane almost touches the postsynaptic membrane, but neurotransmitters are transmitted in between that space. In yeast mating, yeast release pheromones which are identified by neighboring yeast, and a shmoo, which is pointed at on the screen, is formed. This is how yeast mate. In quorum sensing, bacterial cells release a signaling molecule. When that bacteria reaches a threshold, meaning there are a certain amount of signaling molecules in the area, a response occurs. This can be shown in Vibrio fischeri when they exhibit bioluminescence when a certain amount of autoinducers are in the environment. These examples are all forms of short-distance cell communication because the signaling molecules of each example travel a short distance to communicate from cell to cell. The final form of cell communication is direct cell-to-cell communication. This can be seen in gap junctions between animal cells and plasmodesmata between plant cells. These two examples show that signaling substances dissolved in the cytosol pass freely between adjacent cells. Another example of this would be immune cells. Immune cells communicate via direct contact between oligosaccharides on their surfaces. Examples of this could be antibodies, viruses, and macrophages. Another example of this communication is contact inhibition. Cells that are next to each other recognize each other and communicate the message to stop growing. When cells that are next to each other don't get that message, they don't stop growing. This can lead to diseases such as cancer, which is a form of uncontrolled cell growth. These examples are all forms of direct cell-to-cell communication because cells must be touching in order for the cells to communicate. In all forms of cell communication, there are three stages of cell signaling involved in the cell signaling pathway. Number 1, reception. Number 2, transduction. And number 3, response. In reception, a chemical signal binds to a receptor, usually at the cell surface. Number 2, transduction, is when the binding of the cell signal brings about a cascade of reactions. Number 3, response, is when a specific cellular response occurs based on the signal that was received in step number 1. A signal molecule is a molecule that binds to a receptor, causing it to change shape. These signal molecules are commonly known as ligands or ligands, which are small molecules that bind specifically to larger molecules. When the ligand binds to a receptor, the receptor experiences a conformational change, which activates the whole process of reception, transduction, and response. Signal receptors are mostly found embedded within the plasma membrane. There are three types of signal receptors: G protein coupled receptors, tyrosine kinase receptors, and ion channel receptors. The signal receptor receives a signal molecule, which initiates the process of reception, transduction, and response. Now let's apply this knowledge to a diagram. In step 1, the signal molecule, known as a ligand, binds to the receptor. The receptor experiences a conformational change, which triggers a signal transduction pathway, also known as a cascade of reactions. This step is called transduction. After transduction occurs, a response occurs. This response is specific to whatever the ligand is supposed to do. So this is a quick summation of the whole process of reception, transduction, and response. G protein-linked coupled receptors are one of the receptors that I had mentioned before. A GPCR consists of a receptor protein and a G protein. A G protein is a family of proteins that acts as a molecular switch inside a cell. It causes a change in the cell. A GPCR is a membrane-bound protein consisting of 7 alpha helices. So how do these receptors work? First, the signal molecule binds to the receptor on the outside of the cell, which activates the receptor. The G protein is now activated. The activated G protein causes a cascade of further compounds, oftentimes an enzyme, which causes a response. GDP is a molecule that is bound onto the G protein. When GDP is bound on the G protein is inactive. If ATP or GTP is bound onto the G protein is now active. So the G protein system cycles between on and off. When there is a GDP bound on the protein, it's off, and when there's a GTP or ATP, the G protein is active. Now let's get more specific using the example of epinephrine. The ligand, which is epinephrine, binds to the receptor. The receptor undergoes a conformational change and binds to the G protein. This activates the receptor. An alpha subunit exchanges GDP for GTP. An alpha subunit dissociates and binds to and activates the target protein. For epinephrine, the target protein is the enzyme adenylyl cyclase. Now the target protein can relay a second messenger, known as CAMP. Second messengers help to amplify the response. This second messenger continues the cascade of reactions. Next, a response occurs. With epinephrine, epinephrine produces an enzyme that breaks down glycogen to glucose. GTP is hydrolyzed to GDP, which inactivates the G protein once the response occurs. Now let's apply these steps to a diagram. Number 1, the signal, epinephrine, binds to a receptor. The receptor experiences a conformational change. This releases a G protein that is activated when GDP is replaced by GTP. Now the target protein can be activated. In this case, it's the enzyme adenylyl cyclase. Now a cascade reaction occurs, which triggers a response. In the case of epinephrine, epinephrine produces an enzyme that breaks down glycogen into glucose. After this response occurs, the G protein becomes inactive again when GTP is replaced again by GDP. Now let's go over a quick summary of how G protein-linked coupled receptors work. A ligand binds to a receptor. The receptor undergoes a conformational change, which activates it. GDP is replaced by GTP, activating the G protein, which then sets in motion a cascade of reactions, which includes a second messenger, which amplifies the response. This altogether results in a cellular response. The next receptor we're going to be talking about is the tyrosine kinase receptor. This involves 2 ligands binding to 2 receptor proteins. When the 2 ligands bind to the 2 receptor proteins, these polypeptides come together, forming a dimer. The dimer is represented by the purple in the diagram. When these 2 ligands bind to the 2 receptor proteins, this activates the tyrosine kinase section of both. Each protein adds phosphate to the tyrosine tails of the polypeptide, so that's shown by the P's that are attached to the 3 sections on each side of the dimer. The receptor proteins are fully activated when the phosphates are added to the tyrosine tails. The fully activated receptor proteins activate a variety of specific relay proteins that bind to specific phosphorylated tyrosine molecules. These activated relay proteins trigger many different transduction pathways, known as a cascade of reaction, and different responses. 2 different proteins in the body regulate glucose levels. Insulin uses the tyrosine kinase receptor, and glucagon uses the G-coupled protein receptor. The balance of these hormones exemplify the theme of homeostasis with respect to blood sugar levels. I'm going to be discussing specifically insulin with respect to the tyrosine kinase receptor. In a cell signaling pathway of insulin, the ligand, which is insulin, binds to the receptor. Then a cascade of reactions occur where phosphorylation activates the molecules, and a response then occurs which regulates glucose levels. All cell signaling pathways exemplify these 3 simple steps: reception, transduction, and response. All cell signaling pathways are extremely important to life. Regarding tyrosine kinase receptors, when they're inactivated, diseases such as diabetes, Alzheimer's, and cystic fibrosis occur. When the tyrosine kinase receptors are overactivated, diseases like cancer can occur, where cells are growing uncontrollably. The final type of signal receptors are ligand-gated ion channels. Here, ligands bind on the extracellular side of a cell. Protein shapes change and channels for ions open. Here, ion flow occurs. Concentration and charge inside of the cell changes. These type of channels play a vital role in the nervous system, more specifically the axon of neurons, where sodium-potassium pumps are extremely important. At times, instead of binding to receptor proteins embedded within the cell membrane, ligands can diffuse through the cell membrane and attach to signal receptors inside the cytosol or the nucleus of the cell they are targeting. An example of a type of ligand that would use this is nitrous oxide. It is a small gas that helps blood vessels expand and contract and keeps blood flowing and free from clots. Nitrous oxide is able to diffuse through the cell membrane to create a cell signaling pathway. Some hormones also diffuse through the membrane to create a cell signaling pathway. An example of this would be any type of hydrophobic hormone, like sex hormones and thyroid hormones of animals. These hormones bind and activate receptor proteins inside the cell. These activated receptor proteins enter the nucleus, and in the case of sex hormones like testosterone, turn on genes that control male sex characteristics, and this is related to transcription factors. Transcription factors control which genes are turned on, so which genes are transcribed into messenger RNA. Some of these factors are activators, which increase gene expression, while others are repressors, which decrease gene expression. Gene regulation explains the differences between organisms with similar genes. Secondary messengers are small, non-protein, water-soluble molecules or ions. They diffuse rapidly throughout the cell. They help to amplify a message that is created by reception, transduction, and response. Many hormones trigger the formation of CAMP, a form of secondary messenger, in a signal response. They participate in pathways initiated by both GPCR and tyrosine kinase receptors. An example of this would be cyclic AMP, which is what I was referring to as CAMP. Another example could be a calcium ion. In animal cells, increases in the calcium ion lead to the contraction of muscles. In plant cells, increases in the calcium ion trigger responses for coping with environmental stresses. There are two types of signals, hydrophilic or peptide signals and hydrophobic or steroid signals. In hydrophilic signals, the ligand does not pass the cell membrane. The receptor is located on the membrane or embedded within the membrane. They stimulate a transduction pathway, often using secondary messengers. This action is short, measured in minutes or hours. Examples of this could be peptide hormones like insulin, glucagon, epinephrine, neurotransmitters, more specifically acetylcholine. In hydrophobic or steroid signals, the ligand does cross the cell membrane. The receptor is located in the cytoplasm or the nucleus. A combination of receptors and signals act as a transcription factor. The duration of this action is long, measured in weeks or years. Some examples of these could be steroid hormones like estrogen, testosterone, and progesterone, as well as thyroid hormones and nitrous oxide. A final component of cell communication is apoptosis, which is defined by programmed cell death. This is triggered by molecular signals that activate a cascade of suicide proteins in cells destined to die. This is necessary for normal development and differentiation. Disruption of this process can lead to webbed fingers and toes in human hands and feet, immune dysfunction, and certain cancers. Apoptosis is key to homeostasis and is a primary example of cell signaling within various organisms. Cell-to-cell communication is vital for multicellular organisms as well as unicellular organisms. Cells must communicate to coordinate their activities and function properly.